Go. So there it should be recording. Yes, okay, very good. Well, I think this is where we left off the last time. And that was talking a little bit about smooth muscle and several things that we can mention. We already know, of course, that smooth muscle does not have the striations. And that's because the myofibrils are going to be arranged a little bit differently here. So some other things that we find, they have a sarcoplasmic reticulum, but it is not nearly as well developed as it is with both skeletal muscle and cardiac muscle. And in fact, while the sarcoplasmic reticulum is going to store and release calcium and take it back, that's not the only place that calcium comes from in smooth muscle. Smooth muscle cells also have these caveoli. And these are just in-pouchings, if you will, of the membrane, of the sarcolemma, that also store and release calcium. And then another way in which calcium can get down into the sarcoplasm when we want contraction to occur is it actually can come in from outside of the cell. So there are three ways, really, to get calcium elevated in the sarcoplasm of a smooth muscle cell. It can be released from the sarcoplasmic reticulum in the usual manner. It can be released from the caveoli, or it can actually enter from outside of the cell, because there is obviously calcium out in the interstitial fluid. And, of course, then when we want the cells to relax, it can go back into these same areas, back into the sarcoplasmic reticulum, back into the caveoli, or even out of the cell. And to remove calcium from the cell, it's again going to be a calcium pump that operates, and of course, needless to say, that does require ATP. Now, some other things. T-tubules, forget them. You don't find T-tubules in smooth muscle cells. So, the depolarization is going to spread over the membrane surface. And in order to get down into the sarcoplasm, another thing we find are that we do have the gap junctions between adjacent cells, and that can allow some streaming of ions from one cell into the neighboring cells. They have very few mitochondria, and the mitochondria are there obviously to produce ATP, but most ATP production in smooth muscle is anaerobic. So they're relying mostly on glycolysis. Now, of course, one thing that we also note about smooth muscle, it doesn't contract and relax as rapidly as do skeletal muscles or cardiac muscles. So it doesn't really need quite as much ATP as the others do. Some other things that we find. All right, the myofilaments here are a little different. There are, again, thick and thin filaments like we've had before. But the thick filaments, which are myosin, have heads along their entire length. Now remember, where did you find the myosin heads in the case of the skeletal muscle? They just have the two heads right on the end. In this case, the myosin filaments have heads along their entire length. Another thing that is different when it comes to the thin filaments, there aren't as many of them. How many thin filaments were there around each thick filament in the case of a skeletal muscle cell? 6, right. Here it's a lower ratio. And when it comes to the thin filaments, again, they're mostly actin, yes, but they have tropomyosin, again, spiraling over them, covering up the myosin binding sites when it's relaxed, but there's no troponin this time. Instead, we have a couple of other proteins that are going to take the place of the troponin. There is something called calmodulin, and calmodulin is, in essence, a calcium binding protein. I think it's in the next slide. Well, anyway, somewhere. Anyway, I'll just yak. There's calmodulin, which binds calcium, and then it activates a kinase. And between the two, they're going to pull the tropomyosin off the myosin-binding sites on actin and allow contraction to occur. Now, in both skeletal muscle and in cardiac muscle, we saw the myofilaments arranged in parallel bundles. Okay? You don't find that here. Instead, the filaments are in a spiral-like pattern, and that comes across, I think, on this next slide. And I got this somewhere else. It's not out of your textbook. But here's a relaxed, smooth muscle, and there are the myofilaments. And you know what, to me, I like my food images, as you know. What does it look like? It looks sort of like a large sausage has been tied up with butcher's twine, doesn't it? And then, if you pull on the twine, it scrunches up like that. So here it is contracted. But it's like the filaments now are arranged in kind of a spiral around the cell. All right? So there it is relaxed, there it is contracted. And of course, needless to say, when it contracts, it shortens like any other muscle cell, but it just goes about it in a very different manner. Now, when it comes to neuromuscular junctions, we have them, but unlike skeletal muscle where every single cell had its own nerve ending, these don't. Only certain cells, only some of the cells in there will have nerve endings which will stimulate them to contract, and then the contraction passes along in a wave-like manner from cell to cell. So these cells contract slowly and relax slowly in a kind of wave-like pattern. And what happens, of course, with these is that, remember, where do you find smooth muscle? Walls of hollow organs like the GI tract, like in blood vessels, and particularly in the GI tract, especially now that you've had lunch and you're sitting there, and those mixing waves in the stomach, you know, squeezing and mixing things together. Well, they have a kind of peristalsis sort of activity where there is a ring of smooth muscle and there's a longitudinal sheet of smooth muscle, and the two alternate in their contracting and relaxing. So it kind of squeezes to mix and move the stuff through the tube. In the case of blood vessels, it's a circular ring, and it can constrict to, in essence, help raise blood pressure. But in any event, these cells contract and relax very slowly. So they don't need as much energy, and they don't need as extensive a myofibril network as what we found in the other types of cells. So there we are. And as we said, you know, with the gap junctions, they can, move the potential from one cell to another. Some similarities between contraction with other types of muscle. Again, actin and myosin interact. It is a sliding filament kind of mechanism. Calcium is key. Calcium levels in the sarcoplasm increase. They bind to calmodulin. It activates the kinase. It pulls a tropomyosin off the myosin-binding sites of actin, and the myosin heads attach, and then we get the power stroke, and then some ATP attaches, and it detaches from the actin, and then it resets, and it goes around in the same type of pattern. So, again, the myosin is pulling on the thin filaments, and that's what's shortening the myofilaments and causing the muscle to contract. Now, when you want it to relax, what's going to happen? Well, the calcium levels decrease, the tropomyosin covers up the myosin-binding sites on actin, nowhere for the myosin heads to attach, and it just relaxes and it goes back to its relaxed shape and length. You know, some differences. The calcium in this case is not just coming from the sarcoplasmic reticulum, as we said. It's also coming from the caveoli, and it's also coming from outside of the cell. And calmodulin and kinase play a role in positioning the tropomyosin. And the thin filaments, of course, as we said, lack the troponin like we've seen in both skeletal muscle and cardiac muscle. So, you know, there you go. And that's about it. And as we've said, we've gone through this of how it contracts and how it relaxes. And, of course, in order to cause the intracellular calcium levels to decline, you again need the calcium pump, which again is fueled by ATP. And other than that, that's pretty much it. Okay, so there you go. All right, so now we've looked at muscle types and how they operate. So next wrinkle on our little prune is to go to the nervous system. And with that, we've got to switch to another stick. So that will take just a few seconds more to reset. And away we go. Okay, let's see. That here. Okay, now there we go with the nervous system. And this will be, of course, the last system, obviously, that we deal with, but we'll spend some time with it. All right, functions. 3 major functions, and these ought to look like old hat. Because remember, one of the first things we talked about, I think even the first session, I know we did to the second session, and that was talking about a homeostatic loop or homeostatic process. Well, it's basically the same 3 things. You need some kind of stimulus. So we have there, number 1, a sensation where we sense changes either within the body or outside. That is sent off to an area where it is interpreted. And in response to that, we have a response, and that is going off to an effector. And what were the effectors in the case of a homeostatic loop? Either a muscle or a gland. Well, voila, same old thing. Okay? So really, there it is. So we have some kind of stimulus, we have an interpretation of that, and then we have a response. And the response is taken care of by effectors, and the effectors are either going to be muscles or glands, again, like we've seen before. Now, when it comes to the organization, we can start with the central nervous system. And there are 2 big components to it. There's the brain and there's the spinal cord. And those are what we call the central nervous system. All right, and basically this is where sensations are going to go for interpretation and where responses are going to be generated that get relayed out to the effectors. So it's very important in making the whole thing operate. All right, brain and spinal cord. Now, oh, excuse me. Now the rest of it. The peripheral nervous system. And this is nerves that are either bringing sensations into the central nervous system or carrying responses out to the effectors that are outside of the central nervous system. There are 2 divisions. An afferent division, AF, which means into. So in other words, if we're talking about an afferent system, we're talking about sensory impulses coming in. All right, these are nerves that are going to bring information from the receptors into the central nervous system. All right, could be going directly to the brain, but more often than not, they're going to be relayed through the spinal cord up to the brain. Then there's an efferent system. Efferent, EF, away from. You know, like X, meaning away from. All right, these are motor impulses. They're going from the central nervous system where they start, out to the effectors. And once again, what are the effectors? Well, they're either going to be muscles or glands. All right? So there we are. So an afferent division that brings in sensory info, an efferent division that takes it out, motor information, if you will, out to effectors. Now, the efferent system, we've got to make a couple divisions here. I'm going to slice and dice another time, too. All right, we have what we could call the somatic nervous system. Well, this one would in essence be motor impulses that are destined to essentially skeletal muscles. Why? Voluntary. You have conscious voluntary control over these impulses. So if I go running off to get a quick drink of Coke because something I had for lunch didn't agree, well, you know, that's voluntary. All right, so that in essence would be part of that somatic system. The autonomic system, and you might read autonomic as automatic, these are going to be motor impulses that are involuntary, over which you don't have conscious control. So where are these typically destined? Well, what are things we don't have conscious control over? Smooth muscle, cardiac muscle, glands. You know, I don't think anybody's sitting there thinking, oh, I wish the stomach would squirt out some more gastric juice. Okay? Or less, as the case may be. All right, that just takes care of itself. But again, it's motor because it's causing either muscles to contract or it's causing glands to secrete. All right. Now, when it comes to the autonomic nervous system, there are two divisions there. We can talk about a sympathetic division and a parasympathetic. All right, sympathetic, parasympathetic. Is it that boring? Somebody yawn. Now, these are rather interesting. All right, sympathetic is, in general, as we note there on the slide, stimulatory. This is what supposedly prepares us for flight or fight, or however you want to say it. All right, in other words, you're going to stand your ground and fight, or you're going to run away. All right? Parasympathetic is sometimes referred to as what we might call resting / digesting. Okay? Now, what about it? Well, let's suppose that you're sitting here writing your notes as you are, and all of a sudden you hear a disturbance in the hall, and all of a sudden, kind of waddling through the door is a crocodile with its mouth agape. Now, what does that crocodile have in mind, I wonder? It wants a snack. And guess what the snack is? One of us. Maybe more than one if you can get us. All right, now what are you going to do? Are you just going to sit there and say, all right, go ahead? I don't think so. In fact, you really don't even have to think about it. You're going to probably all of a sudden jump up out of your seat and run like you know what to get away from that cussed thing. All right? Now, what's happening as a result? Well, what are you using to run away from the crocodile? Muscles, right? You're going to need muscle contraction. And you're going to need muscle contraction like you never use muscle contraction, because those cussed things can go pretty quickly. All right, now, why do you need that? You need to run away. You need energy. And how do muscles get energy? Well, they oxidize glucose, and they do more efficient oxidation if you do it aerobically. So what's going to happen to the blood supply to your muscles? It's going to increase. Now, if you're increasing the flow of blood to the muscles, what about the GI tract that was just patiently sloshing away with mixing waves and peristaltic waves, you're going to divert a lot of blood from that out to the muscles so you can run away. Now, when you get somewhere a safe distance away from the crocodile, you can then throttle this one back, throttle this one up. What's going on here? Resting and digesting. So guess what's happening now with blood? It's being diverted more away from the skeletal muscles and back toward the smooth muscles and glands in the GI tract. And you go about digesting. So, yes, sympathetic is stimulatory for most things, but it actually inhibits the GI tract. And we'll have more on this later. Parasympathetic is just the opposite. It tends to stimulate the GI tract and not stimulate everything else. Okay? Alright, and as I said, you don't have to think about this. It just happens automatically. You know, you get frightened, and these mechanisms start acting. Alright, if you're resting and relaxing, well, you know, the sympathetic impulses are going to throttle back, parasympathetic ones are going to increase. Alright, now that's simple, isn't it? Alright, so there we go. The autonomic, very automatic, keyed at, you know, things that we don't even have to think about. Now, when it comes to the histology, if you will, of the nervous system, there are two types of cells, general types of cells, that we need to take into account. And we've talked about these in lab. There are the neuroglia, or neuroglia, as some people might say it, and the neurons. Alright, here we have a slide, and let's just cut the lights a bit, showing both types. Here's a neuron, the large cell with that very prominent central nucleus here in the cell body. It has numerous extensions, as you can see. And then you've got these smaller cells surrounding it. Those are, in essence, neuroglia. The neurons are the nerve cells. They're the ones that are actually going to generate and conduct nerve impulses. The neuroglia, on the other hand, are in the supporting role. They're there to help support and protect the neurons. And what about the ratio between neurons and neuroglia? Which one is more numerous? The neuroglia. Far more numerous than neurons. Alright, so as we've said, when it comes to the neuroglia, they're there to support and protect the neurons. And there are four basic types of these neuroglia that we find in the central nervous system. And the first one are what are called astrocytes. Well, astro means star, basically. So, therefore, these have a lot of little star-like projections from them. And they are there to primarily twine around the neurons and the capillaries that you find supplying nutrients to them, and they help to connect the neurons with their blood supply. Alright? So they're there to basically support and help nourish the neurons. Okay? Now, one of my favorite words, oligodendrocytes. Isn't that pretty? Oligodendrocytes. Again, kind of star-shaped, but these are what we would call the myelinating cells that you find in the central nervous system. And they are, as we note, there to produce this myelin sheath around axons found in the central nervous system. Myelin is a phospholipid. And when you have myelinated nerve fibers, they conduct impulses more rapidly than those that have no myelin. And myelinated fibers are oftentimes called white fibers or white matter. Myelin gives things that gross white appearance. What about the ones that have no myelin? They tend to look gray. So when you hear people chattering about gray fibers or gray matter, they're talking about fibers that are not covered with myelin. Okay? But in the central nervous system, it's the oligodendrocytes that coil around the axons, putting on the myelin sheath. And that speeds up their rate of impulse conduction. Now, there's going to be another type of myelinating cell when we get to the peripheral nervous system. We'll see it in a little while. All right, another neuroglial cell are what are called microglia. And these are phagocytic cells. They're phagocytes. They're there to take out debris. They are derived from blood monocytes, and monocytes are one of the 5 types of white blood cells. All right, so just know that they're derived from blood cells, and they're phagocytic cells that are there to take out any debris that might happen to be there in the central nervous system. And then we have the ependyma or ependymal cells. These are essentially a modified, specialized epithelium. They're an epithelial type cell. They are typically a simple epithelium because they're in a single layer. And they vary in shape. Some of them are squamous, some are cuboidal, some are columnar. But one of the things that they definitely have are tight junctions between them. And does anybody remember what the function of those tight junctions was between cells, between adjacent cells? Well, there are proteins in the membrane of the 2 adjacent cells that help seal them together, almost like spot welding them together, so that material can't get between the cells. And because of that, these ependyma, which actually line the brain ventricles and the central canal of the spinal cord, help to contribute to that so-called blood-brain barrier. All right, and remember that is a protection mechanism that we have so that certain molecular substances don't easily get into the nervous system, because the neurons are more easily damaged than other types of cells, and if you've got toxic molecules in the blood, you don't want them crossing this barrier and getting into the brain and damaging the cells. So these ependymal cells help account for that barrier. And what were the ventricles in the brain, for those of you that have had lab already, where we talked about them?
Where the cerebral spinal fluid's made?
Spinal fluid is made, and they're spinal fluid filling. They're fluid-filled cavities within the brain. And the central canal is just a canal, goes down through the spinal cord, and it also is filled with spinal fluid, cerebrospinal fluid. Okay? And if you haven't had lab yet, don't worry, we'll get to it Thursday. We'll have fun with all those things. We did have fun with our brains and eyeballs, didn't we? I thought they were lots of fun. Well, anyway. All right, so now, types of neuroglial cells flying in here. The Schwann cells we haven't talked about yet because they're not in the central nervous system. They're going to be the myelinating cells that are out in the peripheral nervous system. But oligodendrocytes, we've chatted about. Those are the myelinating cells in the central nervous system. They're the ones that lay down that myelin sheath on axons that speed up the rate of nerve impulse conduction, but only in the central nervous system. Microglia, those are the phagocytic cells you find in the central nervous system. They're derived from white blood cells. The astrocytes, those are the ones that help to connect neurons to blood vessels. Now here's one other little thing that you might want to mention, because it does come in handy. Scar tissue in the central nervous system is actually an accumulation of astrocytes. Now, when would you get scar tissue? Well, you would get scar tissue in the central nervous system like anywhere else when there's been damage. Cells have been destroyed, not replaced. Scar tissue gets laid down. If we were out in the skin, what was the scar tissue made of? What kind of tissue was it? Dense, fibrous connective tissue. In other words, collagen fibers. In the central nervous system, it's an accumulation of astrocytes. So if, let's say, someone has had a stroke, their neurons have been destroyed, and neurons don't regenerate. So once they're gone, they're gone. They'll replace the scar tissue, in this case, this collection of astrocytes. And then, of course, the ependyma, those are the ones that line the ventricles, the brain, and the central canal in the spinal cord. All right, so there we go. And here is I think, a horrible diagram, but let's cut the lights and we'll look at it. I don't know why they couldn't give us some actual photographs instead of this drawing, which I don't care for. But anyway, all right, so here we go. Here's an astrocyte. There's a capillary. There's a neuron. It's connecting the two together. And that's one of their functions, of course. Also, an accumulation of astrocytes that serve as scar tissue within the central nervous system. Here we have the oligodendrocyte wrapping around the axons of neurons and, in essence, putting on the myelin sheath. Myelin, remember, is a phospholipid, and it helps to speed up the rate of nerve impulse conduction. We'll find out how and why that works a little later on. Here are microglia. There's one there and one here. Those are the phagocytic cells that are there to remove debris. And then here we have some ependymal cells lining, let's say, a ventricle within the brain. All right, so there we go. Now for the neurons themselves. All right, these are the actual nerve cells. So they're really the functional cells of the nervous system. And they are responsible for generating and conducting nerve impulses from one part of the body to another. All right, what are the parts? There are three major parts to a neuron, and here we have a model of one. All right, first of all, what do we have? We have a cell body right here in the center. Let's open it up, and inside that, what are we going to find? We're going to find a nucleus. All right? And along with the nucleus, there are going to be a number of other organelles. In the nucleus, there could be at least one or more nucleoli. What is a nucleolus or nucleolus made of? It's a nucleic acid, and it's not DNA. RNA and protein, right. Now see, that was easy to reason out, wasn't it? All right, they have a very granular cytoplasm because they've got a lot of organelles in here. Yeah, they're mitochondria, but there is a rough endoplasmic reticulum. And in the case of neurons, the rough endoplasmic reticulum, Nisel bodies. And, needless to say, if it's rough endoplasmic reticulum, what is it likely to be involved in producing? Protein. Okay? And, of course, what was it that made rough endoplasmic reticulum have that rough appearance? Ribosomes, right. All right. Now, very simple, isn't it? All right, there we go. Now, there are also in here neurofibrils. They're there to help reinforce the cell. Well, here they are on this half. All right, so there are the neurofibrils running throughout. And here, of course, would be the Nisel bodies, or Nisel substance, as it's sometimes called. Okay, so there's the cell body. Now, from the cell body, we can have dendrites. Now, there may only be one in some cases. In other cases, like in this model, there are multiple dendrites. They are extensions of the cell body. So these things that you see projecting out from the cell body, those are dendrites. Now, like the cell body, Nysyl substance is found in the dendrites. So dendrites will also have nysyl substance. They'll also have, of course, neurofibrils. Now, when it comes to nerve impulse conduction, nerve impulses can start on either the dendrites or the cell body, and then spread down the next part. So in other words, if it started on a dendrite, it's going to spread down to the cell body, and then from the cell body to the next part, which we're going to call an axon, and we'll look at it here in a bit. If it starts on the cell body, it goes down the axon. It does not go out to the dendrites. It's only a one-way conduction, in other words. All right? And so here is the axon. So if I stimulate this dendrite, the impulse passes from it to the cell body and down the axon. It does not come from the axon up. It doesn't go from the cell body out to a dendrite. It's only a one-way conduction. All right, so there we go. And the axon, the old-fashioned word axis cylinder, but let's just go with axon. Now, doesn't matter how many dendrites, there's never more than one axon, only one. It starts here in an area called the axon hillock, and it can go, well, it can go a long distance. There can be some that have axons that are no more than a few millimeters long. Others, they may be a meter long. So neurons range from being among the smallest cells in the body to the largest, simply because some can have extremely long axonal processes. All right, axons, no nysyl bodies. All they've got in them, neurofibrils. All right? Now, another little tidbit that we need to mention, and that is something that no neuron is going to have, and that is it lacks a centrosome. Now, why would they not have centrosomes? What is the function of a centrosome? When do cells need centrosomes? When they divide. Well, guess what? If neurons don't have them, mature neurons, fully differentiated neurons don't have them, what about their ability to divide? They don't. They can't. That's right. They've lost that ability. All right? Yeah, they would have had it early on, but by the time they're fully differentiated, they've lost it and they can't divide. So what happens if you destroy a neuron? You just lost it. It's gone. It's not replaced. They don't regenerate. All right. The axon, as we said, originates in the axon hillock, and it then continues where its terminal end. It can branch into a number of what we call collaterals, and the collaterals can branch into something called telodendria. And we'll see that term again on another slide. All right? So axons can give several branches called collaterals, and then the collaterals can branch into telodendria. And at the end of the telodendria, we can find something that should warm the cockles of your hearts. At the end of each telodendrion, there ought to be a synaptic knob. What's that for? Well, that's where it connects with another cell like the muscle, or in the case, it may connect with another nerve. All right, on this model, these are telodendria of other neurons, and terminating here on the surface in synaptic knobs. All right? So an impulse might come from another neuron to this one. And then this one could send it off to something else, either another neuron or a muscle or a gland. All right, now, another thing about synaptic knobs. We've talked about them before. What's inside a synaptic knob? There were little vesicles in there. Remember what those were? synaptic vesicles. Remember those? Well, I should hope you would. I haven't forgotten, even though I'm getting old and senile. Now, what's in the synaptic vesicle? What was it when we were talking about neuromuscular junctions? There was a particular neurotransmitter in there, right? What was it? Starts with an A. Ah, say it louder. Acetylcholine, right. And there can be others. All right, but there you go. All right, now that's simple, good heavens. All right, so there we go. And as we've said, the axon is collateral, terminate in these fine filaments called telodendria, and they have the synaptic knobs, and the synaptic knobs contain neurotransmitters, and the one that we've talked about thus far is acetylcholine. And axons contain mitochondria, neurofibrils, but as we said, they have no nisal bodies in them. All right? Now, some other terms related to axons. And how many axons would there be on a neuron? One. Right. There's never more than one. Axolemma is the membrane around the axon. Axoplasm is just simply the cytoplasm inside. All right? So there we go. That's pretty simple. Okay. Now, here we have a diagram. Let's cut the lights a bit. All right, here we have a neuron. So there's the cell body. There's the nucleus in here. And there are a number of dendrites, which are extensions of the cell body. Here is the axon. So there's the axon hillock where it leaves the cell body. And this one has Schwann cells on it. And Schwann cells are the myelinating cells that you find in the peripheral nervous system. All right, what were the myelinating cells that we saw in the central nervous system called? Oligodendrocytes, yes. All right, and here we have an axon branching into two collaterals, and then each collateral has a number of these telodendria at the end. All right, little fine branches of the axon there at the terminal end. And then what were these little enlarged areas called here on the end? Synaptic knobs. And within the synaptic knob, there would be a collection of synaptic vesicles containing neurotransmitter substances. All right, now that's pretty simple, isn't it? Now, one other little tidbit to point out, and then we'll take a look at this model too. There's a Schwann cell, there's one, there's one, there's one. Look at this. They wrap around the axon, but there's a little gap between them. The gap in the axon between the Schwann cells is called the node of Ranvier. Okay? Node of Ranvier. The gap in the myelin sheath between the Schwann cells. Node of Ranvier. All right, now let's take a look at this model. And so here we are. Here's our cell body and the dendrites. Here's the axon. That is one of the Schwann cells there. Here's another. And right here, the little gap between the Schwann cells and the gap in the myelin sheath would be a node of Ranvier. And the way these grow, they actually wrap around this just like you were taking electrical tape and wrapping it around a wire. Okay? And the purpose of the myelin, as we said, speeds up the rate of impulse conduction. So these white fibers, the ones with the myelin, conduct impulses a lot faster than the ones that don't have it. And we'll find out why. We'll get to nerve impulse conduction next time. All right, so there we go. Nerve fibers, axons, and its sheaths or coverings, if it has any. All right, and we talked about the myelin sheath, the white matter, And fibers are called gray matter if they don't have myelin. Okay? And there are a number of those, obviously, in the body as well. All right, now, fibers. And they get connected together to form nerves, and we'll get to that here in a bit. Just another diagram there again, a neuron, dendrite, cell body, there the myelin sheath again. These would be Schwann cells and the node of Ranvier, which is the gap between the Schwann cells, and thus an area where there's no myelin. All right. And as we've said, the Schwann cells wrap around the axon as they grow, putting on that myelin. And other than that, I think we've said all that stuff, so let's just skip the rest. Yeah, there's node of Ranvier. Okay, there we go. And as we said, with the myelin, it increases the speed of transmission. It also helps to maintain and protect the axon. Schwann cells can also play a role, as we'll see, in axon regeneration, too. We'll get to that in a bit. Now, let's talk about neuron classification. Two ways to classify neurons. One is according to their structure. And that's largely dependent on how many processes they have. The second one is by their function. Well, what's that going to be? That's typically going to be either sensory, motor, or what we could call interneurons that connect sensory and motor neurons. All right, we've got that in here in a bit, but I'll just go ahead and mention it now. So when it comes to function, what are the three categories? Sensory, motor, and interneuron. Okay, between the neurons, literally. Now, structure. All right, first one we've got are what we could call unipolar neurons. And sometimes you'll also see pseudo-unipolar or unipolar. Basically means the same thing. Now, one process. One process. So if we're going to do this, there's our, bless you, there's our process. There's our cell body. And here's the process. And so this first area is what we might call the trigger zone. All right, and that's where stimulus can be first applied, and then it conducts down to the axon. But it basically has one process that functions as both a dendrite and an axon. Just one process. That's it. Now, where do we find these things? Well, sensory neurons are typically going to be unipolar neurons. The cell body of these sensory neurons is located in a dorsal root ganglion. Now, we know what the word dorsal means, right? What does that mean? Toward the back. Now, ganglion. A ganglion is an area outside of the central nervous system. An area outside of the central nervous system containing neuron cell bodies. Okay? An area outside of the central nervous system containing neuron cell bodies. And I don't think we have our spinal cord model up here, but that's all right, we'll see it. These dorsal root ganglia are on either side of the spinal cord. They're not part of the spinal cord, so they're not in the central nervous system. They're in large areas toward the back, just outside of the spinal cord, containing the cell bodies of these sensory neurons. So the cell body is located in this dorsal root ganglion. And these are unipolar neurons. And in fact, the largest cells in the body are these unipolar neurons. Because you're going to have some where the cell body is up here in the cervical area, and the axon reaches all the way down to your big toe. Very long axon-type process, about 1 meter in length. Okay? There you go. They're sensory neurons. Now, what could be simpler than that? Well, bipolar neurons, of course. Now, what does the prefix bi mean? Two. Two processors. Tripping over my chair. How nice. All right. Now, what about these? Well, here's our cell body. All right. There is one dendrite. And there is one axon. All right, now the dendrite, yeah, you can have some branches out of here, that's fine. And the axon can certainly end up with collaterals and telodendria down here on the end. That's fine too. But how many processes are actually attached to the cell body directly? Just two. One dendrite, one axon. And of course, don't forget the nucleus there in the center. There you go. That's it. That's bipolar. Now, where do you find them? A lot of sensory organs have these. You find them in the retina of the eye. Okay, the retina of the eye is going to have bipolar neurons in it. You find them in the inner ear. You find them in the olfactory mucosa, you know, where odors are detected in the superior nasal meatus, you know, those kinds of areas. You're going to find bipolar neurons. They're almost always involved with senses. All right, and then we come to the multipolar neurons. Well, that's like the model. Multiple dendrites, but how many axons? One. Okay? Never more than one axon. But multiple dendrites in this case, and these are typically found in both the brain and spinal cord, and in fact, probably most of the neurons you find in the central nervous system are multipolar neurons. All right? And those are the ones that we commonly think of when we think of neurons. But there are some of the others, of course. And here's just a little diagram comparing them. Unipolar, you know, there's the single process, there's the cell body. All right, the trigger zone up here, the axon. Here is the bipolar with a single dendrite and a single axon. And then here we have the multipolar, multiple dendrites, and one axon. And there it is. Okay? Now, it's pretty simple, isn't it? You can't miss that. If everything were that simple. Now, functional, we already talked about sensory, motor, or inner neurons, right? Those are the three categories. And a lot of times, yeah, if they're sensory, they're usually unipolar neurons. We've already mentioned that. So, on we go. Now, enough of that. I don't think we need that. Now, let's say a word or two about nerves. Well, what is a nerve? Well, it's basically a bundle of axons, is really what it is. It's a bundle of axons with its coverings. Now, a nerve will typically contain both myelinated and non-myelinated axons. It will also contain, most of the time, most nerves are what we call mixed nerves, so they will have both sensory and motor fibers, or axons, in them. So you find a mix of myelinated, non-myelinated, sensory, and motor. All right? The interneurons, those go back in the spinal cord and brain. All right, so there we go. And these extend outside, of course, of the central nervous system and extend out to either receptors in the case of sensory neurons or effectors in the case of motor neurons. All right? Pretty simple. And when it comes to the fibers, well, they're typically grouped as one of the four. All right, if we're talking about general somatic afferent fibers, well, what would afferent mean? What kind of information would they bear? Coming in, afferent coming into the central nervous system.
Motor?
Sensory. That's sensory info. All right? Now, somatic, well, that's probably out in things like the skin and the muscles. Okay? Now, general somatic efferent fibers. Efferent means what? What kind of information? It's going out, so it's motor. All right? Now, somatic, motor, skeletal muscle, most likely. All right? That's probably where they're going. Taking information to skeletal muscles to get them to contract. All right? General visceral afferent fibers. Visceral, what does that refer to? What are viscera?
Your organs?
Organs. All right, so these would be sensors and or receptors in the organs. All right? You know, like sensors that detect blood pressure, sensors that detect oxygen levels in the blood, so on, pH of the blood. All right, those would be kind of visceral. And what kind of information is that? Is it sensory or motor? If they're afferent. Sensory. And then general visceral efferent fibers.
Motor.
Efferent.
Motor.
Motor. And motor going where?
Out.
Out where? All right, now if it's visceral, is it likely to be voluntary or involuntary? Yes, involuntary. So what would be the effectors? Those are likely to innervate or stimulate smooth muscle, cardiac muscle, or glands, right? All right. And in fact, when we're looking at these, they're probably part of that autonomic system. Now, the autonomic is motor, they're motor neurons, but they're going to those involuntary types of effectors, like smooth muscle, cardiac muscle, and glands. All right? And so when you have a nerve, if you cut through a nerve and look at the kind of fibers in it, you're going to probably find examples of all four of these. Okay? There aren't many where you wouldn't. Okay, and of course, when it comes to some of these, some are myelinated, some not, and there you go. And some have more myelin than others. All right, so there we go. Now, physiology. All right, regeneration. Well, it's very limited what can happen here. As we've said, when it comes to neurons themselves, if you damage the cell body of a neuron, and it doesn't matter whether it's central nervous system, peripheral nervous system, it's probably going to die. And if it dies, that's the end of it. Nothing else happens. If you damage an axon in the central nervous system, well, that's permanent as well. Nothing is going to repair. The only thing that really can regenerate are myelinated axons in the peripheral nervous system. All right? when it comes to regeneration, it's only possible with myelinated axons in the peripheral nervous system. And then, it's only possible if the Schwann cells remain active. So there have to still be some active living Schwann cells. If there aren't, then even that isn't possible. What about regeneration in the central nervous system with axons? Well, even if they're myelinated there, curtains, the oligodendrocytes are not capable of doing this. Only the Schwann cells can. All right, now, if we go to the diagram, and I think that's better really than the text slide that we had prior. All right, here we have the neurons. These would probably be back in the central nervous system. And here are the axons extending out, in this case, to a skeletal muscle cell. All right, it gets broken. It gets cut. What's going to happen to this distal part? Well, that's going to die and degenerate, completely gone. All right? Now, the proximal part of the injury is going to be capable of growing a new axon if we still have live active Schwann cells. Okay, if we destroy them all, forget it. But if there's some still living here, they're able to then cause axon growth, more Schwann cells produced, and it can actually re-establish the connection to the muscle. Now that will take some time, obviously, but it is possible. And that's about as far as we can go with regeneration in the nervous system. Okay? Because if you were to damage the cell back in the central nervous system, forget it. If you were to damage that cell body, regardless of where it was, that would be the end of it. Because remember, what very essential organelle is missing from a neuron that would enable it to replicate? Centrosomes. Right. They don't have them, so they can't replicate. You damage them, that's it. They're just history. Now, what could be simpler than that? Not much of anything. Now we get to nerve impulse conduction, and I think you've suffered enough. We don't really have quite enough time to get into that, so we'll save that for Thursday. And you'll find it's lots of fun. Now, one thing you'll notice when it comes to nerve impulse conduction, there are going to be a lot of similarities here to what we discussed with muscle contraction, because we're going to have depolarization of a membrane, and that's going to propagate, and there we go. So, I mean, you know, moving ions around, changing voltage, piece of cake. Right? Well, I think, as I said, we've suffered enough for today. So we were very efficient in getting through all that stuff in record time. And I'll kind of stop the video. I'll try to get that up on the page. I'll put it on the web page. And I'll try to stick it up, this afternoon. I have to transfer it over to the computer first.